Ladies and gentlemen, good morning. Welcome to this webinar for Percheron Therapeutics. I think we still have a few folks joining us here, so while they're coming on board, I'll just maybe try and provide a little bit of context to today's webinar. I'm James Garner. I'm the Chief Executive here at Percheron Therapeutics, and I'm joined by my colleague, Dr. Catherine Clary, our Chief Medical Advisor, who's kindly joining us from Princeton, New Jersey. And as I think everybody will be aware, we reported in December of last year top-line 6-month data from our international randomized placebo-controlled phase 2b trial of abicursin, ATL1102, in Duchenne muscular dystrophy. And of course, the results of that study were, to say the least, disappointing. And at the time in December, we really just had the data in our hands for a couple of days. So all we could really do was just report the facts of the matter. But we undertook at the time to go away and analyze the data to really understand in depth what had been happening in the study and to report that back to shareholders. And we undertook to do that by the end of the first quarter, in other words, by the end of March. Well, we still have a couple of points of fine detail coming through, but I think it's probably fair to say we have 95% of the information we're going to have out of this study. And I think it's enough that we can pretty confidently draw some conclusions and determine some answers, at least about the things that have answers. And so we thought it'd be timely to come back to shareholders and to share those results in a spirit of transparency with you. I should say that our intention here today is really to try and provide a reasonably definitive analysis of the study. We don't plan to come back and say a great deal more than we're going to say today. Obviously, if we find some major new insight or some very pertinent new data point, we'll find the right way to share that with people as appropriate. But otherwise, the goal of today's webinar, in one sense, is to close the book on this particular trial. There remains, of course, an open question of the future of ATL1102 and whether we continue to take that program forward. And that's a question we're not quite ready to answer today. We plan to answer that question probably in March. But at least today we wanted to talk about the Duchenne study. So in terms of format, Catherine is going to present some data for about 20 to 30 minutes or so, and we'll then turn the floor to questions from the audience. As a reminder, there is a Q&A button at the bottom of your Zoom window, so you can ask questions by hitting that button. We will, from time to time, need to just very slightly reword questions or to merge very similar questions together just for clarity and for consistency and to avoid repetition. So please understand if your question gets sort of very lightly adjusted on the way. If we run out of time to answer everybody's questions today, we'll find another forum where we can perhaps try and cover those separately. The slides that we're going to present today have been announced to the ASX. They're available there and via the company website. And we are recording today's session, so we will try and make it available via the company website after this webinar, both for your review and, of course, for the many shareholders who couldn't make it in person today. Finally, I'd just say, obviously, there's a great many things going on in the company at the moment, and our focus today is really about the trial data. And I know there's other topics of great interest to shareholders at the moment. For example, had a few shareholders asking us about things like in-licensing activities. We're not really planning to talk about those kind of topics today. We think we can do better justice to those by talking about them separately. We really want to focus today on the trial results, and so that's going to be the focus of today's discussion. So with that said, I'm going to put the slides up on the screen and turn the floor over to Catherine to talk us through.
Great. Thank you, James. Hi, everybody. I'm honored, actually, to be able to present these data to you today. As you may or may not know, I joined the company in June of last year. And since that time, I have been very involved in the oversight of the study, working closely with the study team as well as the CRO. And I must say, when I came in, I was quite impressed with the rigor with which the study was being conducted. So next slide, James. I think James is going to be doing the slide forwarding. There we go. All right. So just to give you a little the background story of the trial, which many of you may know already, and that is that Antisense Therapeutics had completed an open label trial, small phase 2A pilot study of ATL 1102 in 9 boys with non-ambulant Duchenne. And this was done at a single site, Royal Children's Hospital in Melbourne, which, as you know, is a major treatment center in Australia. It was a single arm study designed primarily to look at safety and tolerability in these boys of this age. The performance of the upper limb was measured, though. That is the primary outcome measure that is required by regulators when you're doing studies in Duchenne muscular dystrophy, because we wanted to see what kind of efficacy signal we got with the study. And it did show a very favorable result at the end of 6 months. There was an average change in the total PUL-2, it's called the PUL score of actually a slight improvement of 0.9, which is quite unusual because if the boys were not getting any type of treatment, they generally would be deteriorating. So on the basis of this study, which we felt very positively about, the company originally thought about doing a phase 3 study, which would be a registration trial. However, in talking with the FDA, and we do have an open IND at FDA for Duchenne muscular dystrophy, based on the toxicology studies we had completed to date, they did not want us to go higher than 25 milligrams until we had completed a 9-month toxicology study in non-human primates. It also would have been a very expensive phase 3 study to perform. So in late 2021, the strategy was changed. The company decided they would do an international phase 2B study. It would reduce time, it would reduce expense, but if positive, there was the potential for an accelerated approval based on the severity of the disease and the potential for a biomarker. And this was a randomized placebo-controlled trial. And what that means is patients were randomly assigned to one of 3 groups. And this is what's really required by regulatory authorities. The PUL-2, again, was the primary endpoint for regulatory reasons, and because it is the best measure that we have about the functionality of patients with DMD. The European agencies, by the way, also require this. And we conducted it in 13 hospitals in 5 countries, which meant that there was a very diverse sample, different clinical care characteristics across the sites. But otherwise, it was very similar. So next slide, James. And then a little bit of background on Duchenne, which you probably know. Duchenne is a devastating disease. I have personally met and worked with many boys with Duchenne and their parents, and it is very tragic. As you know, it's an X-linked genetic abnormality. It means that the patients can't produce enough dystrophin, which is necessary for normal functioning of muscle. This then causes chronic inflammation. And eventually muscle weakness, which starts in the legs, and the larger muscles moves up into the arms, and eventually, of course, causes the boys have to become wheelchair bound. And lastly, it affects cardiorespiratory functioning, and that is actually the cause of death. You see here that the life expectancy typically has been in the 20s. More recently, due to the use of steroids and some of the other there's not a lot of agents for it, but some of the newer treatments, patients are living a little bit longer, but it's really quite devastating for the families to see this deterioration occur. Next slide. Now, this was our study. We divided it it was designed in 3 phases. The first phase included a patient population of genetically confirmed DMD. So we did their genetic testing. They were 10 to 17 years of age, non-ambulatory, couldn't walk more than 10 meters without assistance. They were on stable steroid use. We did that because steroids are the standard of care now in DMD, and we wanted there to be not to be differences between the boys in terms of their steroid use. And then they had to have an entry score of PUL of greater than or equal to 2. One would mean that they were really deteriorated, and there was a problem with the not our study, but a different study related to that. It's harder to show a change. The boys were randomized to one of 3 groups, as I mentioned, placebo, 25 milligrams or 50 milligrams, and there were 16 per group. We had originally planned to enroll 45. We did enroll 48. Then we had a 6-month blinded treatment period, and this was the primary portion of the study because it was the only placebo-controlled portion of the study. At the end of that, the patients who'd been on placebo were re-randomized to either 25 or 50. And then the boys who'd been on drug continued on drug. So the second 6 months, everyone was on drug. And then after the year when the patients were to go off drug, there was a 4-month follow-up period for safety. And as you know, we got our initial readout in December 2024. And we were looking at PUL in the primary endpoint. There's some domains of the PUL reflecting shoulder, elbow, and arm and wrist, the MyoGrip and MyoPinch, respiratory function, quality of life, safety, and tolerability. And we looked at week 25, and then secondary endpoints would be the next 6-month period and the 4-month period. So next slide. Now, I've talked about the PUL a little bit. It's been the primary efficacy endpoint of the study. And you see on the left, you see the patient's arm, and you see that there's these 3 levels, which are called domains of the PUL, and there are different levels of functioning. So if you think about the arm for a moment and the shoulder, patient needs those muscles to be able to lift their arm over their head, to comb their hair, to put on a shirt. If you think about the middle level, which is more elbow, it's about being able to lift your hand to your mouth to eat. These are just some of the functional components, but there's a very standardized way that this test is done with a physiotherapist. And lastly, the distal level, which is the use of the fingers and the wrist, and very fine motor control and typically what is seen in the disease is that the shoulder level begins to lose functioning first, then the middle level, then the distal level. The thing about the PUL, and this is what I wanted to point out, and actually many of the functional endpoints, but the PUL, if you look at it and think about it for a moment, let's say a boy came in, he's 13 years old, he's very tired, he's had a long day, he's emotionally upset. These parameters, even though they're done in a very consistent fashion with a physiotherapist, can be somewhat subjective because they do require effort on the part of the patient. This is one of the issues with the measurement of something like the PUL, is that the sensitivity is a little bit variable. Nevertheless, it is the best thing we have right now for measuring the functioning of the upper limb in DMD. Secondary endpoints were the MyoGrip and MyoPinch, and their proprietary measurement devices that have been produced by the Institut de Myologie in France. And MyoGrip measures a patient's grip strength, and MyoPinch their pinch strength, their finger again, they're assessed by trained personnel, but these are actually little machines that measure these. Next slide. All right, so let's get into the top-line study data. So with randomization in a trial that's randomized, the hope is that because the patients are randomly assigned to treatment group, that will sort out the differences between them. They're not chosen because a physician feels they need something, they're randomized. And generally what you want to see is you want to see that the treatment groups at baseline are well balanced. And indeed, we really did see this. So as you see, 17, 16, and 15 patients across the 3 groups. When we look at these slides and graphs in the future, what you will see is that the placebo is on the left in orange, the ATL 25 milligram dose is in blue in the middle, and the ATL 1102 50 milligram dose is in the dark blue on the right. So just quickly looking at this, you see that they were similar mean age, 13 or so, similar weights. There was a difference in the age of diagnosis, a slight difference, and that was that the placebo patients had been diagnosed when they were a little bit younger, and consequently also took steroids. They'd had a mean duration of steroid use that was a little longer because they'd been diagnosed earlier. However, in looking at this statistically, these differences were driven somewhat by outliers and were unlikely to substantially affect the outcome of the study. And so, again, if you look at the PUL scores, very, very similar at baseline, which is what you want to see. Next slide. All right, here's the sort of takeaway top line results. The overall PUL2 measurement at 25 weeks, primary endpoint. Now, when you look at this slide, you'll see again placebo, 25 milligrams, 50 milligrams, and you'll note that the changes were 1.4, 1.8, and 1.6. Those may look a little bit different numerically. However, with statistical testing, which you do to show that the results were not that we wouldn't have seen this result due to chance. These were actually really identical from a statistical perspective. So no difference between them. I mean, it almost looks like placebo's better, but really the takeaway is that there was no difference between the two groups. We also did a pre-specified mixed model regression analysis, which takes into account not only that last measurement at 25 milligrams, but all the measurements throughout the course of the study. And it showed a very similar result. No statistically significant difference between the two groups, either one of the two groups and placebo. Again, a disappointing result because this is the primary result of the study. Next slide. These are the domains that we talked about before, the high level shoulder domain in the middle, the mid-level, and then the distal level wrist domain. And once again, you see very similar although they don't look completely similar numerically, they're close enough that they really are, there's no statistically significant differences. There's a bit of a hint in this, and another slide you'll see that with the ATL 1102 50 milligrams, you see a slightly better result on the distal or level domain. However, again, this could have been due to chance, but we did see it in two different, you'll see next on the myopinch. And we can go to that next slide. And again, on the MyoGrip, MyoPinch, you see on the MyoGrip in particular, 50 milligrams looks a little better. But again, when you apply statistical testing to it, it is not better. So again, there's a slight, but I'd say, but definitely non-significant trend to improvement in the hand function at the 50 milligram dose. And in any study, you can always look at, there's many different outcomes, and sometimes you'll see a positive outcome on one particular measure that's not the primary measure. Generally, regulators and physicians looking at these trials are going to look at the consistency of the results across everything. And unfortunately, here we did see a negative result. All right, next slide. Safety. I mean, obviously, safety is extremely important to us. And similar to what was seen in the phase 2A study, ATL 1102 was really generally safe and quite well tolerated. It's actually unusual not to have any discontinuations due to adverse events in a study, but there were none in this particular study. All the patients stayed the course and stayed on the study. The thing to note here, there were very few serious adverse events that they were related to the actual disease itself, not the study drug. However, what you do see at the very bottom, if you'll put the laser on that, James, that the AEs or adverse events related to study treatment, very few in the placebo group, and then more in the 25, more in the 50, sort of suggesting that there is a pharmacological effect. This is what you generally would see as the doses increase, that you would see more adverse events as the doses increased. But overall, very, very good. And as you know, the primary adverse event that's been seen in all studies with this is our injection site reactions. Next slide. Since we received the top-line results, the Percheron team over the last month, 6 weeks, has really been looking at all the data, trying to figure out, doing some sub-analyses that were not pre-specified, really trying to figure out what has really been going on with the study. And there's actually several questions that we wanted to answer. The first is that sometimes when studies fail, it's because the placebo group does better than expected. So what we wanted to do was look here at other studies that were also in non-ambulatory DMD boys to see how their placebo results looked. And what this slide actually shows, there's two, the most directly comparable studies are the Lelanto study of pamrevlumab, and then the HOPE-2 study, the Capricorn study of CAP 1002. And then Percheron is on the right. So we obviously didn't have a 12-month result, but if you look at the decline at 6 months, Capricor to Percheron did have a somewhat lower placebo response. However, if you look at the bottom line and you look at 12 months data, and then actually if you extrapolated our minus 1.4 at 6 months, it would become 2.8, which is right in between the 2.1 and 3.7 that's seen with the other two studies. So what we've concluded is that actually our placebo group was behaving certainly similarly. It was in between kind of the results of these, but it was in the ballpark of what we might expect to see of a placebo in a study. Another question that we had is the drug biologically active? And so if you look at the next slide, we did a number of PK and PD pharmacodynamic effects to determine that. So first of all, from a pharmacokinetic perspective, looking at ATL1102 concentration, that's on the left. What you'll see is placebo 0. Good thing. There wasn't supposed to be any ATL1102 in placebo. So that was good. And then you look at 25 milligrams, 593, and then 1577 for 50 milligrams. So again, what you would expect, we're giving a higher dose, there's a higher concentration in the plasma. That meant that the drug, certainly from a pharmacokinetic perspective, was what we wanted to see. And I'll show you a little result. I'll show you a comparison with the phase 2A study on that later. Then, as you know, our primary target to decrease inflammation is the CD49D positive T cells, and the hope that our compound would reduce that inflammation. And interesting result here. So if you look at the left, with placebo, it increased. All right, so that's it. They do go up and down to a certain extent, but we certainly didn't see any reduction. 25 milligrams, kind of just on the cusp, not really a lot of change. But then you look at the 50 milligram and you see quite a significant decrease in those cells. So again, suggesting that there is a pharmacodynamic effect, a biological effect on the target that we were trying to hit. And then lastly, on the right, most studies of DMD look at serum creatine kinase. It's an enzyme that exists in muscle cells. And when muscles are damaged, they become leaky and they release creatine kinase into the blood. So what you see with effective drugs in DMD is that you see decreases in creatine kinase. And again, this is perfectly aligned with that theory. I mean, placebo showed minor decrease. And by the way, creatine kinase is a somewhat variable marker. It can jump up and down a little bit. 25 milligrams, we saw a 15% reduction and then 18% with 50 milligrams. So again, it appears that ATL1102 was engaging its target in a dose-dependent fashion. And then lastly, next slide. Lastly, we asked the question, so what happens with placebo patients who've been on placebo in the first 6 months, and then they move to the drug? Because remember, they're re-randomized at the end of that 6 months. And what you see on this is a little bit of a complicated slide, but the peach placebo box, these were patients on placebo the first 6 months. Go to the right, and you see what happens once they switch over to 25 milligrams. And to 50 milligrams. And while there is a bit of a reduction in the week 37 on the 25 milligram, it's really not significant because there were differences at week 25. Don't see much at all with 50 milligram. Similarly, a similar kind of pattern with when you look at the two active doses, when they weren't switched, they were just continued. And we didn't really see a different trajectory to the disease during that second 6 month period. So really looking at this data, and we have about 39, I think we have about 19 patients. There's still some patients well, all the patients are now terminated, but because we terminated the study, they terminated at different time points. So, but 19 patients had completed at least 37 weeks of treatment, and this doesn't really show any trend towards significant or decrease in worsening as patients moved away from placebo. Next slide. All right, so a lot of questions I understand about the phase 2 study. Why is it that the phase 2 study looked the way it did? The phase 2b study looked the way it did. So let's just go through the two studies. First of all, the study populations were broadly similar. Obviously, the phase 2a study had a small sample size. Once again, remember that it was a single center study and everyone was on drug. And what that means is the patients and their families knew they were on drug. The investigator knew they were on active drug. That in a disease like DMD and many diseases, it really arouses a lot of hope and expectancy, sort of the hope that the medicine will work. That can actually drive a little bit of response. It's kind of an unintended placebo response contained within a drug response potentially. The phase 2b study, it's very similar age, weight, PUL2 scores. If you look at, and so really what we saw was with PK and CD49D, we did see pretty similar pharmacodynamic markers. But what the difference was, if you go down to PUL2, that line, the change in the PUL2 is, again, to remind you, was 0.9. It really it's considered stability. It wasn't dropping in the open label study, but we did see drops in both our study drug as well as the placebo in the phase 2b study, and the same with MyoGrip and MyoPinch. There is a little bit difference in the methodology that this was calculated here if you had eagle eyes and notice that the numbers were slightly different, but it just allows us to do like-for-like comparisons with the way they were calculated in the phase 2a study. But again, pretty similar, but we saw a more positive result in the phase 2. So I think I've explained a little bit about hope and expectancy, but let's look at a couple of other things that can help us. Next slide. Unfortunately, it is the fact that drugs which can appear promising in open label exploratory studies, for some of the reasons that I mentioned, do sometimes disappoint in confirmatory randomized controlled studies. And I've talked about pamrevlumab before. They had a nice looking, open label study, went on to a randomized study, and it failed. So they stopped the drug. I've seen this many, many times. I'm a psychiatrist by training, and in doing many different clinical trials across depression and anxiety disorders, which always are placebo controlled. Unfortunately, a lot of studies failed because of the placebo, and they after a while, companies got really good about sort of training investigators to not be too promising, but placebo can be a very powerful drug. And then one other example that we have from the DMD literature in this case is idebenone, which was Puldisa. Again, they were looking at respiratory endpoints, and look at the study on the left, beautiful. Their phase 2a, phase 2b study, beautiful, really hugely positive result on respiratory function. But when they did a big phase 3, it wasn't positive. So again, this is not an uncommon result, and always very disappointing, not only for the companies who are doing these studies, but also for the patient communities who follow these drugs very, very carefully. Next slide. So that's basically the data. So just to conclude a couple of other things, we did look carefully at not only the PK, but also the manufacturing of the drug, the storage, to see if there were any temperature excursions, administration deficits. We did not find any of that. As I said when I opened, the study was conducted very substantially in accordance with the protocol, with rigorous methodology and great oversight of the sites by the CRO and oversight of the CRO by Percheron. We saw that the drug is measurable in plasma and in a dose-dependent fashion. It causes reductions in the CD49D lymphocyte counts and causes dose-dependent reductions in creatine kinase. So in other words, the drug did work as expected on biological parameters, including pharmacological activity. But unfortunately, these results did not result in clinical efficacy greater than placebo that was measurable with the functional endpoints that we needed to use in the study, the PUL2, MyoGrip, and MyoPinch. The placebo group, as I mentioned before, was performing broadly, similarly to placebo in other studies in this population. So as we think through, why did the study not work? Well, we know the drug was pharmacologically active, but it's possible the dose was too low for the activity to meaningfully affect the course of the disease. Remember, we were constrained on dosing by the FDA only because we did the study outside of the US where we were able to use a 50 milligram dose. But there's hints, and I say only hints, that perhaps a higher dose might actually have been better, but we really couldn't use that from a safety perspective. The drug was pharmacologically active, we know that, but it's possible that the mechanism, which is inhibition of CD49D positive lymphocytes and dropping those, is just not enough to meaningfully affect the course of the disease. And that there are other disease mechanisms, and there are, that cause continued clinical deterioration. And remember, these are boys who've been ill for a while. They were 13 when they came into the study. So they've been experiencing substantial muscle deterioration for some time. Our last hypothesis is, and I've kind of alluded to it, that while the drug was pharmacologically and clinically active, the actual measurement instruments were too insensitive to detect a treatment effect of a drug versus placebo. So the ATL 1102 treated groups behave differently in some respects from that phase 2A study where we only saw 25. And I think, again, it probably reflected study design, single study, greater expectancy effects, perhaps just chance. But we can't tell for sure if it's 1, 2, or 3, but we think these are the 3 most likely causes. So I will conclude there, and I know we are happy to take questions. Thank you.
Catherine, thank you very much indeed for that very, very helpful walkthrough. So we have a couple of questions that have come in. We're going to move straight on now to questions. Just a reminder for everybody who's attending the webinar, there is a Q&A button at the bottom of the screen. If we can ask you to use that to send through your questions, and then we'll try and sort of take them in the order they come, more or less. So, Catherine, we've had a question about MRI, and of course there was we used MRI as an exploratory endpoint in the phase 2A study. Saw some promising indicators, some increase in lean muscle mass, some reduction in adiposity. Wasn't an endpoint in this study. Maybe you could just sort of talk through the thinking there, and perhaps a little bit about how regulatory agencies regard this sort of data.
Sure. I mean, MRI is a very interesting endpoint, and it's something that the community of academics and DMD treaters are very interested in, because of similar results to what we saw in our phase 2A study. MRIs are complex. They have to be very carefully calibrated. If you think about a multi-site, multi-country study, some of the countries didn't have high technology sites. You really have to calibrate those results. The methodology for doing the MRIs across in order to get a good reading. We knew that we would need to use the PUL and these other endpoints. And we also know that regulators, while they're interested in MRI, have not yet accepted it as a primary endpoint in the study. They're very expensive and very complex, and that is why we decided not to pursue MRI in this particular study.
Thanks, Catherine. We've had a question about the post-trial access program that can be run. Back in, I think, September or October, we said that we're going to provide drug after patients had completed treatment in the study, which of course is a pretty common thing in these kind of trials. And I think some folks are trying to sort of square that with the ultimate results of the study. Why were the investigators keen to do that, given the eventual lack of efficacy? So again, maybe you can comment on post-trial access programs and and sort of what the investigators were seeing at the time.
Sure. So first of all, yeah, post-trial access programs now are very standard across studies, particularly where there's been a placebo. Patients have had exposure to placebo. It's felt that they should be able to continue on the drug. Of course, we did not know the results of this study when we started our post-trial access program, but we felt it was the right thing to do. Our investigators felt it was the right thing to do. They did not know what their patients had been on. They knew that at the end, everyone was on drug. Because at the end of the 6 months, everyone, but they didn't know what patients had been on placebo. And actually, if you look at the results, some patient within a group of 16 or 17 patients, some patients are going to be doing very well and others much less, because we were looking at just means. So there's the hope that they would continue to be getting drug and do well or do better. And the other thing is that there's really so little. This was free drug being supplied to them, and there's so little else that they can get access to, that I think the investigators told us they wanted to give their patients the greatest chance of continuing to not deteriorate as quickly.
And we've had a sort of almost a CMC question here, a chemistry manufacturing controls question. There's a question about what exactly is the placebo in this trial? And of course, those of us that live and breathe clinical trials bandy these terms around every day. But one of our shareholders has asked, what exactly do we mean when we say a patient's on placebo? So yeah, perhaps you could just comment on sort of what a matched placebo looks like, what the placebo-treated patients were doing.
Sure. Yeah. Yeah, no, absolutely. So the patients are, our dosing schedule in the study was a sub-Q injection with alternating places once a week. So what the placebo is an inert substance, also a liquid that is injected, just similar to the drug, so that the patients won't know whether they're getting a placebo injected or a active drug injected. I can't tell you exactly what the substance was, but as I said, it's inert. It's not biologically active at all.
Thanks, Catherine. Switching from CMC to statistics, questions come in to ask really whether the study was too short. Would a longer study with a longer period of time on drug have given us a different result? I wonder if you can comment on that.
Sure. Yeah, it's a really good question, because those of you who've been sort of following the DMD field know that many companies now are doing much longer studies than the 6-month study. However, if you look at our results and just look at the lines that are going on with placebo and the drugs, I don't believe that a longer study would have produced a result. There just wasn't a difference between the three groups in this particular study.
Thank you. There's a question here about ATL1102's shelf life, which I realize might be stretching your comfort zone here. I can address that if you'd like, or you're welcome.
Why don't you go ahead?
Sure. Well, look, I think the long story short answer to this is the drug is very, very durable. This is a pretty robust drug. It lasts for some number of years. And it's not a drug which requires very, very controlled storage conditions. Some drugs have to be stored in the refrigerator. Some drugs have to be frozen until they're used. Some drugs are incredibly fragile and sensitive. This is not one of those. This is a pretty robust drug. And one of the things we did as part of our investigation into this study was to recall some of the vials of drug from the hospitals and check them to make sure they haven't been damaged in transit in some form. And we really found no indication they had. They seemed to be, as good as the day they left the factory. And the PK data that Catherine presented, the concentration of the drug in the placebo group, the 25 milligram group, and the 50 milligram group, really looked exactly as we would expect it to, which really suggests that the drug is intact, that it's getting to patients, and it's getting to hospitals in exactly the way that it should. So we're really confident that there aren't those kind of issues. We didn't have an issue here with the drug product in the trial. It is sometimes an issue. Some of the drugs we work with are incredibly fragile. This one is pretty robust, and we don't think that was any part of the result here. Um, so, Catherine, a question that's come through about other ways of measuring efficacy, particularly, of course, in the non-ambulance setting here. And I think you made the point very well that PUL2 and MyGrip and MyoPinch, they all have an element of subjectivity in them. They all have an element of variability in them. And so the question has come through: have we got better instruments for this? And indeed, is there anything coming? And I know you've attended a few workshops in recent months with groups like PPMD about this, and
Yes.
you've been right at the center of these discussions. So, wonder if you could maybe comment a little bit on just how people are thinking about solving some of these
Sure.
challenges.
Sure. I mean, it's a really great question, and it's also a really exciting area of research because and it's a lot of it is driven by organizations like PPMD and families. And I mean, the mothers of boys with DMD are the most passionate, committed group I've ever met. And they realize that these are not ideal instruments. And so, what they've really taken upon themselves, and a lot of researchers as well, is to figure out other types of measurements that aren't as subjective, things like video monitoring of the boy in his natural setting. So, it's not a test that's being taken, it's just how is he functioning in that natural setting. And there actually is a video monitoring tool which has been developed by a mother and from PPMD, but a whole group of researchers that is in the process of getting certified by FDA. Probably still, though, will not be a primary endpoint. It could be a very strong secondary endpoint. There's also a lot of digital tools. If you can imagine, everybody's now wearing, rings and Fitbits and all kinds of things. So digital monitoring of movement and things like that is another area that's being looked at. Unfortunately, it takes a very long time to get these tools certified properly and validated such that the FDA is convinced that you're actually seeing a real result. But it is an area of research, and I think we will see over the next few years, we will see some of these tools coming into use and being accepted by regulators.
Thank you. Thank you. A question that's come in around the possibility of interference with the results. Is there any possibility that a rival company may have sabotaged the study? Obviously, perhaps a little bit of a sort of forensic attitude to things, but an understandable question. Again, perhaps, Catherine, you could comment on just how these studies work and the rigor with which they're conducted, and just how hard that would be.
Yeah, I mean, clinical trials are really one of the most rigorously evaluated, monitored activities that we do. So I think I mentioned before, we had a global CRO. They've got significant policies and procedures to ensure drug accountability, that the drug's being measured properly, that the patients are coming in for their visits at the right time. They look at the doctor's notes versus the CRF that we're looking at to see the clinical data to make sure that it all matches up. And then there are also audits. So sites get audited. The CRO itself gets audited. Our CMC that's making the manufacturing sites are audited. So there's lots and lots of audits. And all these are required in order to submit to a regulatory body. I think it would be very difficult. I mean, honestly, I have not heard. I've seen instances where perhaps a drug gets substituted at the, it gets mixed up. I saw a trial where that happened, where actually the placebo was it. But I've actually never seen something where there was sabotage like that. So I don't think that's would have happened. No.
Understood. We've had a couple of questions just about the mechanics of how the drug was administered. Obviously, AT1102 is a weekly subcutaneous injection. And of course, the matched placebo is administered the same way. And a few people have been asking, was that done in the hospital? Was it done at home? Was there any difference between how that was done in the phase 2B study versus the phase 2A study? Again, maybe you could just touch on how this works, the training that patients go through and so on.
Sure. Yeah. So so again, like you said, it was a weekly sub-Q. There was some training involved. It was when the patients were first injected, they were injected at the clinical site, whether that be a hospital or, but it was a clinical site with clinical personnel who taught the caregivers how to inject the site. And whenever they came in for a visit that was around that time, they would get it injected at the site. However, they were trained so that they could the caregiver could inject them at home. The patients needed to rotate the injections because, as I mentioned, there are injection site reactions in some patients, not all, but in some patients. But yes, it could be injected at home, and we were thinking about how that would work, what that formulation would look like if we were to continue on into phase 3 and eventually commercialization.
We had a question about just the investigators' response to this, and in particular, of course, Professor Thomas Voigt at Great Ormond Street Hospital in London, who is the lead investigator on the study. Folks have just asked what the feedback from investigators were, and were they surprised?
Yeah. I mean, when we got, that was a very busy weekend when we got our results, because of course we got on the phone with Dr. Voigt, who's been our principal investigator, is our principal investigator, and has been very, very involved in the study, as well as informing all of the investigators about the results of the study. I would say to a person, they were disappointed because they understand how difficult this disease is, and there's such a need for new treatments. I wouldn't say that they were surprised, not because they had any negative feelings about ATL-1102, but because it has been so difficult to prove efficacy in this disease. But yes, they shared the disappointment of our company and of the patient community.
Absolutely. We've got time for maybe 2 more questions here, and I might say one that I might just take the liberty of addressing at the end, but perhaps the penultimate question is around just the costs associated with closing down the trial. And we put out in our most recent financial statements, we estimated there was about $6 to $7 million of costs to sort of come through the tubes here. And I think we've had 1 or 2 questions to the effect that, well, how come, given the study terminated in December? Catherine, maybe you could just say something a little bit about the sort of backlog in invoicing here, and just where the study actually is operationally, but also just a little bit, I suppose, about how CROs are always behind.
Sure. Yeah, well, I mean, if you think about it, yeah, the last patient actually was it took a while for some of the patients to get in for their termination visit, because we wanted to make sure, do safety checks, and we had a case report form for the termination visit. So once the patients have stopped coming, then there's a cleanup process that occurs between the site and our CRO, making sure that everything matches, all the data is accurate. That then goes to the CRO. They have to clean it, do an analysis. That takes some time. That goes to a statistical group. So there's a lot of activities post the patient stopping to come into the study that still keep happening. And then the company is often not billed after that. Since CROs, every study I've ever run, we have to basically keep on the backs of the CROs to get things done quickly. It's just, it's a big operational challenge to do that. So our team, and the team here at Percheron is great, have really been working closely with the CRO to try to get things done as quickly as possible. One example of that is that there will be a clinical study report that's always written at the end of a study. It's quite detailed. It has all the data. We are going to be doing an abbreviated clinical study report, which will be quite a bit shorter. It will have safety data. It will have a little bit of efficacy data. And that's because we're not going to continue with this study. We're not going to continue with this indication. So we're trying to do everything we can to try to save a little money to get the study closed.
Thank you. I'm going to maybe just in closing to address one question myself. And there's been a few questions as we've been talking about the future for ATL-1102 and what this trial result means for other potential uses of the drug. I think one thing we've been clear on for many years as a company is this drug has multiple potential use cases, multiple potential diseases that it could be used to address. And I think people will be aware that just because a drug perhaps doesn't work the way we hope in one disease, it doesn't necessarily rule it out in others. So what is the future for ATL-1102? Well, I'm going to slightly handle that question at the moment and say that is something that the board is still thinking through very carefully. And we're taking some expert advice on this. We're talking to a range of people to really try and think this question through methodically. And there's a very fine line for a pharmaceutical company in this position between, on the one hand, throwing the baby out with the bathwater and discarding a potentially promising drug, but on the other hand, not wanting to invest good money after bad. And I think we're trying to find exactly the right balance between those two extremes on behalf of shareholders. We don't have all the answers to that yet, and I think that's something we hope to be able to talk more about next month, as we just sort of work through these considerations. But again, the drug has been tested in a range of other conditions, muscular dystrophies like limb-girdle muscular dystrophy, neurological diseases like multiple sclerosis. We had some very promising preclinical data last year in autoimmune epilepsy and so on. So we're really thinking carefully about that, and we'll have more to say about that next month. One thing I would say, and I think this perhaps segues into some conversations we'd like to have with shareholders in the near future, is that one way or another, we don't think that ATL 1102 can remain the centerpiece of the story that the company puts in front of investors. And I think this was something that was clear to us as soon as we saw the results in December, that the company, in order to attract investor interest, in order to build the company back to where it should be, in order to get the share price up and to draw investors back into the stock, I think is going to have to find something new to talk about. Now, whether that is in addition to ATL 1102 or whether it's instead of ATL 1102, that's the thought process that I think we need to go through over coming weeks. And as I say, as we sort of reach conclusions there, as we start to get that kind of feedback, and as we work out the best way forward, we'll be sharing that with shareholders in real time, as we have been with the study results. But for today, I think that concludes our webinar. I just want to thank Catherine for taking up a good chunk of her early evening to talk through this. I think hopefully this has been helpful for all of the folks on the call to just understand what we've seen. We wanted to, as I say, give you as much clarity as we possibly can about the study results. Realize there are sometimes questions that occur to people afterwards. I just say again, please feel free to make use of our contact email address, info@percherontx.com. You can ask questions also via our website. We really want to make sure we help shareholders think through this data and share everything we can about the results we've got. So hopefully today's webinar is part of that dialogue rather than the end of the story. But equally, hopefully it's given people some clarity about what we've seen in the study. So I'm going to conclude the webinar there, say thank you very much again to Catherine, to the team at Hack Digital for organizing, and thank you to the many shareholders who've joined us today online. And as I say, we'll be sharing more progress over coming weeks. Many thanks again. Bye now.